Okay, so chapter 13 is very much related to chapter 12 when we talk about mitosis. In chapter 13, we are also talking about nuclear division, but in this case, we are talking about the process of meiosis. So a lot of what we focus on in this chapter is going to be distinctions between mitosis and meiosis and how the processes are different. So we want to pay attention to that as we go through this. We are also talking about, in this case, sexual reproduction, whereas mitosis focuses on asexual reproduction. So the other thing we'll be talking about in this chapter is the variation that is introduced through the process of meiosis, nuclear division. So the first thing that we have to get a handle on, and we're going to be coming back to this time and time again throughout the semester, is that these processes of mitosis and meiosis deal with passing down genes, or the DNA molecules. When we talk about genes, we are simply talking about DNA. So the nucleic acid that is in the nucleus of a cell, that is DNA. And DNA is equal to genes. When we talk about inheritance of genes, and you're going to talk about this over and over again throughout this course as well as in genetics, we're talking about passing down the DNA. And when we talk about DNA, we're talking about the chromosomes. So the chromosomes that we talked about in chapter 12, this is the DNA. This is what we're talking about here, and we're talking about passing that down from one generation to the next. Specifically, this comes into play in meiosis because we are talking about the fact that genes are passed down from one generation to the next through these reproductive cells, and in this case, the gametes, which in the humans are the sperm and the egg. So, meiosis, one way that meiosis is different from mitosis is that we actually have 2 rounds of division, not the 1 round like we saw in mitosis. So there are 2 rounds of division that occur in meiosis. And so whenever we are talking about meiosis and talking about the divisions, we have to distinguish. It's absolutely essential that we distinguish whether we are talking about meiosis 1 or meiosis 2. Different things are happening in each of these divisions, and so we have to distinguish whether we're talking about the first round of division or the second round of division. So in meiosis, we start out with our what are called homologous pairs, and we're going to walk through this in just one second. The first round of meiosis actually causes these homologous pairs to separate. So one homologous pair would come from mom, the other homologous pair would come from dad. So we'll walk through that in just one second. The second division in meiosis separates the sister chromatids. When we go through this division, you are going to see that it looks a lot like mitosis. Just in mitosis, we did the exact same thing. We separated sister chromatids, and that's exactly what's going to happen in meiosis 2. So the process of meiosis 2 looks a lot like mitosis. Okay, so let's talk about this. We're going to go through the phases of meiosis, going first through meiosis 1 and then through meiosis 2. So meiosis 1, just like mitosis, has 4 subphases. So we call those prophase 1, metaphase 1, anaphase 1, and telophase 1. So these are sounding really familiar to what we talked about during mitosis. The distinction is we have to specify that we are talking about the first division in meiosis by calling it meiosis 1, and then prophase 1, metaphase 1, anaphase 1, and telophase 1. So a couple of things are going to happen here. We are going to duplicate the chromosomes. That happens, remember, in S phase of interphase. So you got to go back and think about the cell cycle on that one that we talked about back in chapter 12. And this time we are going to be dealing with homologous pairs, and I'm going to walk you through what a homologous pair is and how we get there in 1 second. And we are going to end up with 2 daughter cells at the end of meiosis 1. So we start with what's called a parent cell, And at the end of meiosis 1, we have 2 daughter cells. All right. So prophase 1. So it looks a lot like prophase of meiosis, where a couple of things are happening. That spindle apparatus is forming. So remember, the centrioles are going to move to the poles of the cell. So those are our centrioles. And that nuclear envelope is going to disappear. So you can see we're starting to get lots of holes in that nuclear envelope, and it will eventually be gone by the end of prophase 1. One thing that is unique to meiosis 1 that we did not see in mitosis is something called crossing over. So if we're going to look more specifically at crossing over. So here we have a homologous pair of chromosomes. Remember, this is 2 chromosomes, and they are duplicated. So we really have 4 sister chromatids here. 1, 2, 3, and 4. But we have 2 chromosomes that are in their duplicated form. One of these chromosomes you got from your mom, and the other chromosome came from dad. So what can happen during prophase 1, and this only happens during prophase 1 of meiosis, is where the non-sister chromatids exchange pieces. So this is a molecular interaction between a chromatid of one chromosome and a chromatid of the homologous partner. So you see here we have exchange between the light blue piece and the teal piece. And what happens is you end up with chromosomes that contain pieces of the other chromosome or portions of the homologous partner. So this is what leads to variation in traits. This is one way that we introduce variation in traits. This is why you don't look exactly like your mom or exactly like your dad, but you're kind of a combination of the two. So this crossing over is the first way that we introduce variation of traits in meiosis. Notice we did not see this in mitosis because, remember, mitosis gives us a genetically identical copy of the parent cell. So mitosis results in cells that are genetically identical. Meiosis allows for variation, and that's where this crossing over comes into play. All right, so once we get to metaphase 1 of meiosis 1, it's going to look a lot like metaphase of mitosis, except we now have these homologous pairs lined up along the equator. So we still have our spindle apparatus formed here with our centrioles at the poles. We get our chromosomes lining up along the equator. And here, during metaphase 1 of meiosis 1, we get the second variation, or the second way that we introduce variation in traits. And this is called random arrangements, which we're going to take a closer look at on the next slide. Okay, so we're going to talk a little bit more about this random arrangement now and how that leads to variation in genes. So basically, this can occur because the way that the chromosomes line up along the equator is completely random. So when we line these homologous pairs up in meiosis 1, in metaphase of meiosis 1, we can have various combinations or various ways that they are arranged along the equator. So for example, these cells here, they all have 6 chromosomes. They're all duplicated because they've all gone through S phase, but they are in their duplicated, so they're in their duplicated form, but we have 6 chromosomes in each of these cells. So let's say that the light blue ones, for example, came from your mom, and the dark blue ones came from your dad. All right, so the way that these chromosomes can line up along the equator give us varying combinations of chromosomes in our daughter cells. So if they were to line up like this in this first picture, where we had all 3 of mom on the top and all 3 of dad on the bottom, our daughter cells that we produce would all look like this. So these might be mom, mom, mom. These would be dad, dad, dad. Now, it doesn't have to work out that way. So here we have a different arrangement. We have dad, mom, mom on the top, and we would have mom, dad, dad on the bottom. We could have this arrangement, or we could have this arrangement. So you see we have lots of different ways that these chromosomes could line up on the equator, which would result in very different daughter cells. So we have 1, 2, 3, 4, 5, 6, 7, 8 different arrangements of or different daughter cells that we could get from this one 6 chromosomed containing cell. Remember, your cells contain 46 chromosomes, so you could get many different more arrangements of cells or many different combinations of chromosomes based on the random arrangement. Alright, so once we get through metaphase, we get random arrangement. So we have had 2 areas where we have introduced variation. One occurred in prophase. That was the crossing over. And then we had the random arrangement in metaphase. And so now we are ready to continue with meiosis 1, and we enter into anaphase. So anaphase is where we start to pull those chromosomes to opposite poles of the cell through the shortening of the microtubules. And in this case, we get duplicated chromosomes that are pulled apart. So in anaphase 1, it is key to know that we are separating homologous pairs. So one chromosome from mom would go to one end, one chromosome from dad would go to the other end. One chromosome from dad might go to this end, and one chromosome from mom might go to this end. So it is the homologous pairs, the mom-dad partners, that are separated in anaphase 1 of meiosis 1, and that is very key to remember. Alright, once we get to telophase 1, so remember that is nuclear division. This would be immediately followed by cytokinesis, which is the cellular or the cytoplasmic division, and that is what is represented here. So in this picture, cytokinesis has already occurred. We end up with 2 cells, 2 daughter cells, that are what we call haploid. Remember, haploid means that they have one of each type of chromosome. So our parent cell in this case was diploid. In this case, we had 2 of each type of chromosome. So for each chromosome, we had one from mom and we had one from dad. In the case of the daughter cells that we make at the end of meiosis 1, both of these cells are what we call haploid. We only have one of each type that comes from either mom or dad. Okay, so when we enter into meiosis 2, the key is that we do not go back through the entire cell cycle. So we do not go through interphase again. We do not go through the synthesis phase where we replicate the cells again. We go straight from meiosis 2 into meiosis 1. So the key thing to remember is that there is no DNA replication between the 2 divisions. So here we are at the beginning of meiosis 2 with our 2 haploid cells that we made at the end of meiosis 1. We're going to carry these cells right through meiosis 2 with the same 4 subphases that we've been talking about. The result in this case is going to be 4 haploid cells. Okay, so I'll walk you through how we get there. And in this case, the other key thing to remember in meiosis 2 is that we are separating the sister chromatids, where in meiosis 1 we separated homologous pairs or homologous partners. Now we are separating sister chromatids. All right, so prophase 1 is, prophase 2 is very similar to prophase 1 of meiosis 1, or even prophase of mitosis. So we get a new spindle apparatus forming with our centrioles going to the opposite poles, and the chromosomes are going to begin to move towards the equator, and also that nuclear envelope is going to disappear, go away. All right, so here we have metaphase 2. Our chromosomes are lined up on the equator. Our spindles are fully formed. Our centrioles are in place. The microtubules have grown out, and they have attached to the centromeres of our chromosomes along the equator. In anaphase 2, it is the microtubules have shortened, and it is the sister chromatids that are pulled apart. Okay, this is key. We're separating the sister chromatids in anaphase 2 of meiosis 2. And then lastly, in telophase 2, followed by, again, cytokinesis, we end up with 4 haploid cells. So what we get here at the end of meiosis 2 is 4 cells that are haploid, meaning they have one of each type of chromosome. And I'm going to walk you through an example of this in a second and kind of give you an application for how we would carry out meiosis 2. Okay, so here we have a slide talking about gamete formation in the male, which in the human, this is the sperm. So we're going to talk about sperm formation and how it works as a function of meiosis. So we start out over here with this cell, which is called the spermatogonium. This is the diploid male reproductive cell. So this cell, if we were to look at it, it has, this particular example, has 4 chromosomes. So we know that it is diploid, which means it has 2 of each type of chromosome. So we're going to say it has 2 long chromosomes and 2 short chromosomes. So each of these long ones, you got one from mom and one from dad, and the same thing with the short ones. You got one from mom and one from dad. Now, this cell is going to undergo or go through interphase. So from block 1 to block 2, this cell goes through interphase, where, remember, it goes through G1 and S and G2 phase. So the cell gets bigger and the chromosomes duplicate. So in this cell, in block 2, we still have 4 chromosomes. They are now duplicated. So they're now in their sister chromatid form, but they're still 4 chromosomes. They're just now duplicated. Now, it is this cell, which is called the primary spermatocyte, that is going to undergo meiosis. So from here, from block 2 to block 3, that cell undergoes meiosis. All right. So, we'll call this block 3A right here, and we'll call this block 3B. So in 3A, this cell has undergone meiosis 1. And so, by the way, this primary spermatocyte is still a diploid cell. It still has 2 of each type of chromosomes, 2 longs and 2 shorts. When we undergo meiosis 1, so in block 3A right here, we end up with 2 cells which are haploid. Okay, so 2 haploid cells at the end of meiosis 1. Each of these chromosomes now has each of these cells now has 1 long chromosome and 1 short chromosome. 1 long chromosome and 1 short chromosome. So these are haploid cells. Now from 3A to 3B, we undergo meiosis 2, and we now end up with 4 haploid cells. So 4 haploid cells, 1, 2, 3, 4, and they each have 1 of each type of chromosome. So 1 long, 1 short, 1 long, 1 short, 1 long, 1 short, 1 long, 1 short. These cells, which are called spermatids, eventually develop into fully mature sperm once they undergo differentiation. So sperm cells are indeed haploid, okay? And that's really important for us when we talk about fertilization, because these sperm cells are eventually going to find an egg cell. That's their job in life, that's their goal in life, is to fertilize an egg from the female. Egg cells are also, by the way, haploid, which we're going to talk about in just one second. When these two things come together, they form the zygote, which is always going to be diploid. So we take those two haploid cells that we make in meiosis II and turn them into a zygote, which is diploid. If we had diploid cells from our gametes, that would cause real problems when fertilization happened. When we formed these zygotes, we would have twice as many chromosomes as we should have. So remember, each of these haploid sperm is going to have 23 chromosomes. Each of these haploid eggs is going to have 23 chromosomes, and then our zygote is going to end up with 46 chromosomes, which is the correct number. So gamete formation in the male is pretty simple, pretty straightforward. Each of these spermatids would develop into a fully functional sperm. So from one round of meiosis in the male, you end up with 4 fully functional sperm. That is not the case in the female. Okay, so lastly, we want to talk about gamete formation in the female. And in humans, this is formation of the egg, or what we call the ovum. Okay, so it happens a little bit differently than in the male, but the primary steps are the same. So here we have what's called an oogonium. This is the female reproductive cell. It is also diploid, just like in the male. So in this case, in this particular example, this cell has 4 chromosomes. They are unduplicated right now, so they're not in their duplicated form. And this cell is, again, just like in the male, going to go through interphase, where we go through G1, S, and G2. And those chromosomes are going to duplicate. So this cell, again here, has 4 chromosomes. They are just now in their duplicated form. And this cell is also diploid. This cell, just like the cells in the male, are going to undergo meiosis and go through the 2 divisions. So this would represent meiosis I, this would represent meiosis II. Now what is different in the female is when this primary oocyte undergoes meiosis I, we get unequal division. You can see that one cell is larger than the other cell. So we get unequal cytoplasmic division when we do this in the female. So the cytoplasm of this primary oocyte does not divide equally. We end up with the smaller cell is what we call a polar body, and the larger cell is what we call the secondary oocyte. You can see that both of these cells are haploid. They have 1 of each type of chromosome. Now the polar body serves absolutely no function whatsoever in the organism, and it would eventually go away and just dissolve on its own once and after it divides. So when we undergo meiosis II, so from here to here, we're going to take those cells and divide them again in meiosis II, and we're still going to end up with our 4 haploid cells, which we know we get from meiosis. When we undergo both rounds of meiosis, we end up with 4 haploid cells. Again, we're going to get this unequal cytoplasmic division when we divide both of these cells. And now we end up with 3 of these smaller these smaller cells, which are the polar bodies, and only 1 cell that can serve as the ovum or the egg. All of these cells would be haploid. That's important to remember. But 3 of them are these polar bodies, which again serve no function whatsoever inside the organism, and they would eventually dissolve or just go away on their own. The ovum, this is what we talked about on the previous slide, the ovum would join with the sperm during fertilization to form the zygote. So remember, our ovum and our sperm are haploid, so that our zygote can be diploid. Now we're going to do some activities with this in class so to help you better understand what's happening to all the chromosomes here. So I will see you in class to go through all of mitosis and meiosis so that we can make clear distinctions between the two. One other clear thing that I want you to keep in mind when we talk about mitosis, okay, so when we talked about mitosis in chapter 12, I'm going to write this in a different color actually. So when we talked about mitosis, we talked about that it maintains chromosome number. All right, that was a key thing that we talked about in chapter 12 with mitosis. With meiosis, the key thing is that chromosome number is cut in half. Okay, so let's look at this. For example, if we look at our oogonium here, this thing had 4 chromosomes. We talked about that. If we look at our egg or our ovum here, the number of chromosomes in this cell is 2. So chromosome number has been cut in half. That will happen every single time you go through meiosis. You go from a diploid cell to a haploid cell, and chromosome number is halved. So that is very key to understand, very key to remember that distinction between mitosis and meiosis. I will see you in class.